Hello, everyone. Welcome back to Cells to Cell podcast. This week, we're going to actually be going over the FDA approval processes, not only in America, but the FDA kind of version in other countries like in continents like Europe, countries like South Korea, and we have an interesting little episode for you today. It's not going to be as long as last time. It's not going to be an hour. We realize that's too much. We got to keep things more engaging and more short and entertaining. So we're going to go over short things. Hopefully have more episodes for you guys, and we hopefully you guys like this new setup also. We decided to look at the camera this time.
Yeah. Maybe it feels a little more natural.
Yeah.
And not like you're listening to us talk at a coffee shop, but kind of more interactive. So that's kind of what we were going for. But yeah, like Heem said, we're going to talk about some FDA stuff and some World Health Organization and stem cell therapies across the globe, and how, there have been stem cell therapies being used, in different countries for over 10 years. They've been used for over 10 years for a multitude of different things, and why they have them, but we here in America do not, which is kind of an interesting topic. So go ahead and kind of just tell them about the paper and everything a little bit.
So this paper goes over all the guidelines basically that show why it's really difficult to get FDA approved stem cell therapies in America versus in places like Europe. And it goes over basically the characterization of the entire process from getting the stem cells to administering them. So when we talk about guidelines, we're talking about literally everything. Where these stem cells are being sourced from, how they're going to be used, who is going to be using them, literally the plastic that it's going to be grown in the labs, what devices are going to grow them and expand them, so which incubator is going to be used. And all of this has to be done under very specific guidelines here in America. But in other countries, they actually don't have as strict of laws for the sourcing of the stem cells. So in America, FDA wants to know where the stem cells are even sourced from, what tissue, and they need a lot of data to back that up. And in places like Europe, they don't really have that. They have a organization called the
It's not just an organization in Europe. It's just the international. I had it up and I should have left it.
Stem cell and gene therapy.
it's the International Society for Cell and Gene Therapies. So they kind of govern or set guidelines for how tissue engineering and stem cells kind of should be taken care of, and what steps should be taken, what precursors should be hit, like what you need to kind of have before you can even start testing. And what's funny about it is that even though it's an international society, the FDA still does not kind of fall in line with that, which is, I find, kind of odd, because, for the United Nations, we have the World Health Organization, which kind of helps oversee tissue engineering and stem cell therapies as well. And they kind of are in line with that International Society of Cell and Gene Therapy, but the FDA is still, has like a stranglehold on how much they're asking of these scientists before we can, any stem cell testing can even be done. And I think one thing before we even dive into this paper is just to give a brief overview of what the FDA does, and how let's put cell therapies on the back burner for a second, and let's just talk about normal drugs. So let's take acetaminophen or Advil, for example. With COVID and everything happening a couple years ago, we were looking at all of the different new vaccines, like the mRNA vaccines from Pfizer and Moderna. They were constantly going through these phases, and everyone was talking about, phase 1, phase 2, phase 3, phase 4, all this kind of stuff. And I think it's just important before we even talk about the FDA, what they do and why they kind of do it. So you just kind of start after you discover a drug and you present it to the FDA. You have to apply before you can even start testing. As they review everything, and then once you get approved, you go into phase 1 trials, which is basically a safety trial. You give the drug to a healthy person and you see if there are any side effects or adverse effects, and how you can maybe change that or anything. Phase 2 is when you really get into the testing. So that's when you go into small studies and you go ahead and you not only are continuously check for safety, but you see if there are any results in the sick person that you are trying, or the sick population that you are trying to help or cure. And then phase 3 is just blowing phase 2 onto a whole new level. So you're going from phase 1 of hundreds of people, maybe tens to hundreds of people, whereas in phase 3 you are going
tens of thousands.
into thousands, if not tens of thousands. And there you continuously are checking and making sure that the drug is still safe. You're giving it to a bunch of different people, whether that is sick people of different ethnicities or ages. And it's still very, very strict in making sure that no one's being taken advantage of and that everybody's being kept safe. And then finally, if you pass that phase 3 meeting with the FDA, you actually get to go and market your drug, but you continuously go into phase 4. And phase 4 is continuously monitoring that medicine in patients and across different clinics and across the nation or the world, if you can get it approved there. So just kind of understanding that it's just it's not a simple process, but laying it out simply. When we talk about phase 1 trials and we talk about phase 3 and phase 4 trials, especially with these cell therapies, I know in the last episode I talked about Dr. Arnold Kaplan suggesting in his paper that they throw out phase 2 and phase 3 testing because it doesn't fall in line with how cell therapies are kind of wanting to be administered, or how it doesn't, because cell therapies don't follow the normal drug submission.
So what Nick mentioned with phase 4, there's not a legit phase 4, but it's kind of like seeing like what happens after everything's done. So I'm going to include a graphic for our viewers to see, but there's 2 main things that a company having a new drug or a cell therapy would have to quite literally, through regulation, have to apply for. So the first thing is called an IND. It's called an investigational new drug. So that's you and your team saying, hey, from all of our research that we've done, we think something might work. So now we're going to call on the FDA and we're going to have them work with us. So that's an IND, an investigational new drug. It's in the name. You're trying to investigate whether this new drug is going to be good or not. And then you start doing your clinical trials. So like Nick said, your phase 1 is going to have tens of people just to see like whoever is the really needy population of this particular drug. If phase 1 goes well, we'll try and do phase 2, which actually has more controls and more people in it to see how it affects people who don't have as progressed of the disease. So that'll be more like hundreds of people. Phase 3, then you go into your thousands. And once all of that goes well, throughout that entire phase 1, 2, and 3, you're going to have amendments that you can make to your investigational new drug. So at first, the application you made, your first IND, that is going to connect with your end application called an NDA, new drug application. So your IND happens. That means, hey, FDA, come on and check us out. We'll go through phase 1, phase 2, and phase 3. And throughout those 3 phases, you can make amendments to the IND. And then at the end of it, if it all works out, then you have a new drug application, so NDA. And once you get approved for that application, then you have a new drug in the market, or a new cell therapy, or a new gene therapy. And that's basically the whole timeline of how things go.
And this timeline takes between 8 and 10 years, between finding a new drug and getting that NDA submission accepted to have a drug put on the market. It can take 10 years or more, eight or more years. It's a very, very lengthy process, and it involves a lot of moving parts, making sure that everything is consistent, like he said at the beginning. So one thing I want to say is that not every country is necessarily like this. Mm-hmm. It's important to understand that the United States, there are probably multiple reasons that we could have another whole podcast on why the FDA is so strict on on having these drugs and getting FDA approval is actually so important. But I want to kind of get into the paper a little bit.
For sure.
Because we've given some background. Hopefully, we still need to set up an email so people can send in questions if they do have any questions. So that'll probably be put in the description at the bottom of this video. And if you have any questions about episode 1 or this episode, or the following episode, you should have an email where you can contact us directly and ask us any questions, and we'll try our best to incorporate that in the coming episodes. But let's talk about mesenchymal stem cells and how they're kind of viewed throughout the world and what kind of happens with them in other countries.
So there's this nice figure in this paper that actually shows which countries have approved MSC therapies. The United States has zero MSC approved therapies. The therapies we do have approved are umbilical cord stem cells, which are very pluripotent. They're very rich in viable amount of cells. And other countries, they have approved MSC therapies for indications like subcutaneous tissue defects, myocardial infarctions, Crohn's fistulas, knee cartilage defects, graft versus host disease. So that's just if you're getting an organ transplant and you're having some sort of rejection from the organ to your own body, MSCs can actually be used to help counter that. And even things like spinal cord injury. And it's very interesting because the sourcing of all these MSCs comes from actually a few different types of tissues. There's adipose tissue MSCs, there's bone marrow tissue MSCs, and there's also umbilical cord MSCs. And now that's interesting because the United States has only umbilical cord MSCs, mm-hmm. and that's because there's no difference between this person's umbilical cord and that person's umbilical cord. The cells are so fresh and they're so pluripotent, and they have such good characteristics that they don't have issues that we mentioned in the last podcast, where just when an adult grows up, your MSCs will actually become stuck to your tissue type. So when you take adult MSCs and give them to another person or even give them to yourself, it's really risky because that's not a standardized process. In other countries, it's approved for some people because it's if you have this issue and you want to get it done for yourself, you can do it. But I feel like in the United States, something has to be, if you're going to approve something through the FDA, it has to be applicable for every single type of person here.
Yeah, and it goes into the whole issue of HLA identification and matching. So, for example, if him and I were to, have, say I need a blood, and he was like, I'll give you blood, and our blood types don't match, he can't give me blood because it'll kill me. And that's the same thing that goes for organ transplants. And anybody that knows someone who's going through an organ transplant, you put on a list because you're waiting for, a fresh organ that matches you, your blood type, everything like that, to be given. And even when you match, there's still a ton of complications, especially graft versus host disease. And I think that the interesting part about the umbilical cord stuff is, like you said, that those cells are so fresh. Mm-hmm. They're still so new, and even when you cryo-freeze them and preserve them, I think they're good up to 16 years, I believe, or something like that, or maybe even longer or shorter.
Yeah,
but it's a good amount of time to where the matching doesn't have to be. The matching's a little bit more loose than having to be a perfect match.
Yeah,
you only have to have so many. You only have to check so many boxes before you can say, oh yeah, I can give you this umbilical cord blood and help you. One thing I wanted to actually mention about these different drugs or these different therapies was not only the types of tissue, but also where they're from. So at the top of the list, there are 4 drugs from South Korea, and four stem cell therapies in one country is pretty remarkable in my opinion, especially when it's
2010 and 11 and 12.
2010, 11, and 12.
That's quite advanced.
Three medicinal, MSC
therapies.
3 MSC therapies that really, really help people. There's one in New Zealand, another one from South Korea in 2014. Japan has two, one in 2015 and one in 2018. And then India and just, it doesn't specify, but it just says Europe. So I'm assuming, it's applied all over the place. So, and again, we're in the year of, we're at the end of 2022, and 2018, we're 4 years behind, which we're not necessarily behind in the science, but in getting new drugs on the market that can help somebody, it kind of feels like we're kind of behind a little bit.
And what's interesting is that there's a term called, what is it called?
Pharmacogenetics?
Going abroad for a medical treatment. Tourism. Medical tourism. Medical tourism is a big issue because there are people that are in America and they know that places like Germany have MSC therapies that can help their loved ones. And it turns out to be cheaper also because there's not a lot of regulation around all these kind of things. And when you think about your loved one being in such a serious situation and there's nothing in America legally that you can do about it, the only option is leaving this country, going to somewhere else and getting that therapy and coming back. Now, the FDA in America can't stop you from doing that.
No.
But let's think about our economy. If we want to be a leading country in this world, in science, in industry, in all these different things, it has to be built in America and it has to be given to our people here. There's no reason for a person who's wealthy and who has the money to go abroad to get a treatment. Yeah, you have the freedom of will to do so, but that's taking away the whole point of, America and us being a world leader. And I think that's very interesting, and how medical tourism is a big issue because FDA hasn't approved a lot of these MSC therapies. And I think that's something that definitely needs to change in the next 10 years.
I've met somebody who's participated in medical tourism, and they had stage 3 or 4 melanoma. They flew to France, got some stem cell therapy, and it cured them. It metastasized to their liver, and it saved them. And that's the type of stuff we're talking about when we, the lengths that we can go to with these stem cell therapies. And it's, how many in the United States, melanoma is pretty controlled, but at the same time, like breast cancer still plagues, a very large group of people, especially like triple negative breast cancer, which is deadly. It's sad to hear, but like when somebody gets it, when you hear that, wouldn't you want the doctor to say, we have these new drugs just coming on the market to give you a chance? And I feel like the FDA is kind of holding those back. They're not given like leeway, and I'm not asking for leeway in like the safety aspect of everything, but it goes back to Dr. Kaplan's comment of test them for safety and then just jump straight into stage 4 trials and administer it and monitor very, very closely. And if it's helping, keep it going, and until there's a reason to stop that therapy, keep it on the market. I think the mindset towards these new cell therapies is kind of lacking, in my opinion. But that's also coming from someone in school and excited and learning about it, and not from somebody who's FDA trained and looking at all the safeties. So, it's just an opinion. But at the same time, if I had some sort of cancer, I would want to use the newest stuff that I feel like could give me the best chance of survival. And I think that we kind of need to, push a little bit more.
I think it's just the reason that it's all so delayed in America is because MSCs, as outlined in this paper, they suffer from lack of standardization. We want to emphasize to you that MSCs are in every single place in your body. Your eyes have MSCs, your forehead has MSCs, your eyebrows have MSCs, your nails have MSCs, your hand has MSCs, your knees have MSCs, your heart has MSCs. Not as much as other things, but For example, your heart won't have as many MSCs as your fat because your fat proliferates a lot. There's a lot more tissue in a fat. So sourcing MSCs and having a standardized set of procedures that any hospital in America can follow is kind of non-existent for MSCs because they can be sourced from so many different tissue types. For example, even if I wanted to source it from fat, I can use my belly fat, I can use my leg fat. Does that differ in its ability to heal you? There's a lot of things that go into this, and the only reason that umbilical cord cells are approved in America is because they are very standard. They can be used for any blood type. They're pluripotent. They come from the same place that's after the birth of a child, and there's some standardization to that. So if FDA is going to say, hey, this is approved, then you should be able to get this treatment in California, in Arkansas, in Michigan, in Ohio, in Florida, all the same. But it's not that easy when you talk about MSCs, and how they actually get MSCs is something called cell surface markers. So you want to explain what cell surface markers are?
Yeah, basically I liked how you were talking about it before the podcast.
Yeah.
Was you basically, the way that I've learned about it so far here at Case is through bone marrow. So you have a ton of MSCs in your bone marrow because, like you said, with the fat, bone marrow is constantly proliferating. It's constantly making new cells. So they would usually take it from your iliac crest or like your hip bone, like from the back, and they take out 5 milliliters at once, 5 milliliter aspirations, and what they do is they kind of spin it down, extract the cells, and are looking for cell surface markers. So the way that cells are made is they have something called a phospholipid bilayer that encapsulates everything inside. So all the DNA is inside, all the mitochondria is inside, and everything else that makes up a cell. But on that phospholipid bilayer on the outside, there are protein markers and glycoprotein markers and all this stuff that give these cells some
specific identity.
So it differentiates the cells from other cells. So that's why we have skin cells versus our neurons, or I guess that our fingernails aren't really cells, but you get the point. It helps them differ. So when we look for stem cells, we're looking for, I believe it's 4 specific markers that give them their identity. And what's really interesting is that we were talking about how your stem cells kind of differentiate between even the same tissues. So like my leg, the adipose tissue on my leg versus the adipose tissue on my stomach, or like in the front of like my trunk, they might differ, but they all still kind of have the same makeup. So one that I am aware that exists as a cell marker is CD34. So there are ways to test for CD34 on cell surfaces, and that it's like picking out like a criminal from a crowd. Like if you were assaulted and they brought in 5 people from the street and you had to stand in that behind the glass and pick who was who, you're able to say like, oh, I know that they had blonde hair, I know they were wearing glasses, and I know that they were above 6 foot. So then you can eliminate 3 of the 5, and then you only have 2 left, and then there's other tests and questions that you can do. And that's kind of how we pick out these stem cells, but again, they're very variable. They although they have the same cell surface markers, they can differ.
Definitely, and I think that goes to show you how difficult it is to have a standard set of procedures for something like this, because if your fat from your belly versus your leg has CD34, but then there's a difference between the secondary marker. So for example, the example Nick gave, not only blonde hair but also glasses and above 6 foot, the same way stem cells work. So not only do they have CD34, but they can be CD133 positive or negative, and then they could have another protein also, which are not CD, so they can be things like c-Met.
Yeah.
If c-Met is tyrosine protein kinase, it's a protein that makes you very specific to a certain tissue type, and it is really difficult to identify, and that's why it's really difficult and why we don't see a lot of MSC approved therapies.
And I think it's important to understand that these cell therapies are not like any other drug. A lot of drugs that we take on a daily basis are molecules or groups of molecules, and those molecules have to undergo serious environmental changes in order for anything in that molecule to change and thus give you a different reaction. These cells are living things. They change with what environment they're in, temperature, pH, or how acidic their environment is. It all is it's all variable. It's all ever changing in our bodies when we take them out. You know, what's interesting is I mentioned mesenchymal stem cells in the bone marrow. I just had a lecture yesterday that I found fascinating, and our teacher was talking about mesenchymal stem cells when they're in the bone. So we mentioned that they were parasites last week. So those parasites again are kind of monitoring what's going on in that bone marrow, in the empty spaces of that bone marrow and the blood vessels to make sure that we have enough blood. When you take those out and you put them into a beaker or a plate to test them, a petri dish to test them, they all of a sudden go from being parasites to being stem cells, where you can differentiate them and make them into new progenitors or new specialized cells, and they will divide and proliferate until they have no more space to do so. You put those cells back into a body, into the donor or into a patient, they go back into parasites. They lose what's called their stemness. Their stemness is talking about how they can proliferate and differentiate.
That's so interesting to me.
It's crazy, and it's wild how nature has created the human body, and how we think of cells just like they have one job, they do this. But again, like I said, they are ever changing. They are constantly reacting and making sure that their environment is staying healthy, and if not, they have ways to change it. So it's important to, and again, this whole variability of these cells and how different they can be and how they react is why the FDA is so hesitant on it. So while I have an outcry for these new stem cell therapies to be put onto the market, at the same time, I would never want to push for a drug that would do way more harm than help.
Exactly. False hope is not a good thing.
No,
especially if some of these therapies are going to be hundreds of thousands of dollars. Like, yeah, current treatments are not going to heal you as well as stem cells will, but I think having a lot of profound data, and a lot of it is really good, because a standard set of procedures will always win over having something that you feel like you should do just because it exists. And that should actually wrap up this week's topic.
Yeah, no, I thought that this paper was fantastic. Again, this paper is jam-packed with everything. We should probably start just linking our papers in the description to see if people, if you're interested in what we're reading and kind of want to see what we're doing, or something sounded funny and you have a question for us and you want a reference to a piece of the article in the literature. But like I said, we're going to set up the email. Feel free to reach out to us. We would love to help and answer any questions to the best of our abilities. Again, I want to remind everybody that we're students.
Yeah,
we're still learning. And we're doing this to, bring you guys on the journey with us.
Yeah.
So I just want to say, thank you for the support of the first episode. I know it's not like astronomical numbers, but it did a lot better than I could have ever expected, and I'm so happy for those who watched it.
We hope you like our new setup. We're
it's continuously, it's going to change. It's going to constantly change. I'm just warning you. So, all right, guys. Well, thank you for tuning in to episode 2 of Cells to Sell, and we appreciate you listening. Have a good one.
See ya.